Welcome back, friends. In this video we'll be talking about different type of mRNA splicing. Not mRNA, actually, we have studied about the mRNA splicing because the splicing event that we all know is for the mRNA or messenger RNA. But in this video we are going to learn that splicing event is not only unique to mRNAs, but also we can see protein splicings. So we'll be talking about protein splicing or splicing of proteins. We'll be talking about splicing event of proteins. Now what we know about the splicing, the very basic understanding of splicing is that, so if we are having mRNA, suppose if we are having the mRNA sequences, let's say this is the mRNA sequence, these two regions are exons, and for example this middle red part is the intron, so it is intron, and these two parts are exons, right? So let me write here, it is exon, it is also an exon. Now the reason or the process of mRNA splicing is telling us that we don't require these intron units because the introns are not carrying any gene of our interest which are coding proteins. The protein coding genes are present in exon regions, so we only want exons to be attached together, so we get a whole length of mRNA sequence which will code for totally for proteins, not for any other elements. So this part of the sequence, which are the introns, they are placed between two exons, are also called the junk elements, because those elements are not required for the protein synthesis. Remember, but previously when it is discovered it was called the junk element, but nowadays we know that this is not junk because it plays very important role. Now the goal here is to combine these two things together. So if we combine these two things together, what we get is something like that. So it simply make a structure like this. So we have discussed about various types of splicing. So here it is the intron, and these two are so this is exon 1, this is exon 2. So E1, E2, they are combined together. So what we get is this with E1 and E2, and along with that what we get is this. Okay, so this is the I, exon 1, exon 2, and this is the I, which is the intron. So we get this the result. This is called the mRNA splicing. This is the basic overview of mRNA splicing, and we have studied several type of splicing and different proteins that are involved in splicing in detail. So if you don't see these videos, if you don't know this concept, please go back to my YouTube channel. You will find the video on mRNA splicing. Just watch them. Now in this video we'll be talking about protein splicing. Now there are some other individuals or other organisms, now in those organisms what happens, there are not only mRNA splicing but also protein splicing events occur. That means the splicing means simply cutting of the introns from, or that means the cutting of the segment we don't require for protein synthesis can occur once after the protein is synthesized. So now for example in this kind of individual's body, individual cell, what we get the sequences, we get this DNA sequence, this protein sequence. So let me draw the protein sequence like that. Say this is the protein, this is the exon, this is another exon events which are called extein in this case. In this case we call it exon, but here we call it extein, and this red segment is called the intein. Previous case in mRNA we call it intron, here we call it intein, T-E-I-N, and here these blue segments are called extein, E-X-T-E-I-N. So it is extein 1 and it is extein 2. So we are having two exteins, and on the middle we are having intein. Now this is a protein. I remind you, this is a protein. So it is a protein. Now from where this protein is synthesized, the protein is synthesized somewhere with from the DNA segments. Now we get this protein. Now from this protein we don't require this middle segment or intein for the structural purposes. So as soon as the protein is produced, now remember this type of proteins are not even get the 3D conformation. They haven't get any 3D structure. Just this is the basic amino acid sequences, and simply the secondary structure of proteins, we get this kind of structure, extein both the sides, intein in the middle. Now we don't want inteins for the further purpose inside the cell at that particular time point. So what we'll do, we need to cleave this intein out. So for this region what we'll be doing, we just cleave this part out of this section. So how can this part is out? Now there is a process of this cutting off these inteins out from this segment, and we'll be studying that how this process is done. Similarly, that just like this previous case, so this intein is cut out and exteins are linked together. in this case. Okay, so this thing can happen. Okay, so but the important thing is that, so again let's say here, so what we'll be getting, we'll simply get this thing joined together, and also along with that we get our desired. Yes, so this is the intein, and this is the extein 1, and this is extein 2. So that's how we get our desired product. Now this protein is our desired protein. So the process are very much similar with each other, but the accomplishment of the process and the steps are quite different. Now let's look for the detailed steps of this protein splicing. Now if we draw the structure of how this inteins and exteins are arranged, then we can draw, or we can get the structures. For example, say here, say let me draw the structure. This is another extein, and on the middle we are having the intein sequence. So this is intein, and both these sides we are having extein. So this is extein 1, this is extein 2. Okay, now let's look for the special structure of this kind of proteins. Now when we see this kind of proteins, there is a special amino acid arrangement. Similarly, if you remember, when you discuss about the mRNA splicing, we have seen also specific sequences are there which are triggering the process for the cleavage. And normally for this cleavage or this splicing event, we know that this intein part or intron parts play the important role. Similarly here also for protein splicing, we'll be seeing this intein part playing important roles. What kind of roles? For example, in both this terminal part, which is called the junction point of this attachment, junction point. For example, say this is the star mark, this is the junction point, or the branching point, we also say the branch junction or junction point, from where they just cleave out and join two exons together to get our desired product. So this is a branch point here, or the junction point here. In both these points, what we get, both this intein and extein points, what we get is the serine or cysteine amino acid. So either serine or cysteine amino acid sequences. So this is conserved. This is pretty conserved throughout the evolution. We all know this is a very important observation using bioinformatics studies. Now, as we are having serine or cysteine, so what is the advantage of having serine or cysteine? We are having SH group for cysteine. We are having OH group for serine. So if we are having serine, we are having OH. If we are having cysteine, we are having SH group. Remember, because the side chain of serine is containing OH, side chain of cysteine contains SH, right? Now what is the advantage of having these groups? That this group can be electrophile, so they can be converted into nucleophile, not electrophile, and they can attack the other extein and other sequences. Now here this is the extein, and here this is the N-terminal, and this is the C-terminal of the protein. Because we are talking about proteins, so it is very important to mark the N and C-terminal. As simply just like for the mRNA, it is very important to mark 5' and 3' terminal, right? So here it is the N-terminal, this is the C-terminal of the protein. Now what we get is this sequences. Now the very first step of this reaction is simply for cutting this bridge or between linkage between extein 1 and intein. So this is the first step, and to cut from here intein and extein 1, and then the extein will be held together with this SH bond with this intein sequence. So let me draw the structure. The structure will look like this. So this is the intein, and here it is the extein 2. This is the C-terminal, and what we have here, we are having SH linkage. So with this linkage we are having extein 1 attached. So this is the very first step of the reaction, which is catalyzed. Now this kind of reaction is catalyzed by simply nucleophilic attack, because we are having the OH. Now if we are having this OH, because this OH can be nucleophile, right? So we can have this. So this can act as nucleophile. This can attack here, so that there is a cleavage and we get this kind of linkage. Now after having this linkage, remember here is also we are having another OH, because if we are having a serine here, for example, let's consider we are having a serine in both the direction, both the place, so we are having an OH here. So after this step, what we get, we simply get this part of the extein segment transferred from this link to this link. So now what we get this is our intein, and here we go, this is our extein 2, and here it is extein 1 will be attached with this linkage. So here it is. Now simply this reaction means simply transfer of this extein 1 from this serine to this serine. That's very simple. So as we are having breaking a serine this bond and also making the same type of bond, the net requirement of the energy is 0. So you don't require any extra energy from outside. So again here we are having the transfer of this to this serine. Here it is another serine, and this serine becomes free now again. So after this step what will happen, now we can see that at the terminal, so this thing actually happened at the terminal. So what we have at the terminal, E1 and E2 are in very close proximity. Now what will happen, now here there are different groups that are present in inteins that trigger another round of reaction, catalytic reaction, self-catalytic reaction. So all of these processes that we are seeing, all of the processes are self-catalytic reactions. So they are all self-catalyzed reactions. So remember this is self-catalysis. So you don't require any kind of catalyst in this case. We don't require any enzymes. This is all self-catalytic reactions. And here again, due to this catalytic reaction, this intein will cut out, and then extein 1 and extein 2 will be joined with each other. So after that, the final step is to joining of these two exteins together. So what we get is, here it is, extein 1 is attached with extein 2, and what we get is intein, which is cut out. So this is the result after everything is done. So this is the process of protein splicing. Okay, now what are the important players here? Serine or cysteine in the both junctions. That is very important. Now we call this one branch junction because the reaction starts from this place. So these two junctions are very important, and the amino acid place both junctions are very much conserved. Okay, now this is the basic process of done. I'm not talking about the detailed chemical processes and those arrow reactions. I won't talk about those. But what one thing I want to talk about is a very interesting concept about these inteins, especially, because what happens on all these cases, on all the directions, what they want to make sure is that none of the system which don't require these enzymes at this time can utilize this enzyme. That means simply this intein that is getting degraded from the extein systems, from this whole system, is a type of DNase. This is a potential DNase, and obviously it is a double-stranded DNA cutter. This is a potential double-stranded DNA cutter, and it is very strong. I mind you, it is very strong. So this intein is a very strong DNA cutter. Now what it does actually, whenever this kind of splicing event, protein splicing event is done properly, then we have these intein sequences out of the system. Now this intein act as a DNA cutter, so they will go and cut the double-stranded DNAs. Okay, so let's consider this inside the system cell. Here it is a cell. Inside the cell we are having this mixture of this exteins and inteins. So this red thing is the intein. Now after the cutting event, what we get, simply intein is out and exteins are attached together. So these things just prepared. Now this intein act as the DNA cutter. So here it is a DNA. So let me draw the DNA in green color. So here it is the double-stranded DNA sequence. Now this intein can go and cut this DNA sequence. So what it creates is a double-stranded break. It creates a double-stranded break in this DNA. Now this double-stranded break is very difficult to repair, because remember, we can only repair the genes that are present in double-stranded break if and only if they are having the 2 copy of the same gene or 2 copy of the same DNA, right? And the only way of getting 2 copy of the same DNA is possible in case of eukaryotes, because we are having 2 set and 2 pairs, one from father, one from mother. But for the bacterial cells, they don't have any 2 set of the same gene or 2 set of the same DNA. So what will happen, if this thing happens inside a bacterial cell and inteins are generated, they can go and cleave the DNA, cleave it to give that double-stranded break. Now this double-stranded break cannot be repaired inside those bacterial cell, and eventually the cell will die. So the result for the bacteria, if the cell is a bacterial cell here, suppose this is a bacteria cell, now the end result of this process is death. Here comes a very important concept about inteins. Now you know that inteins, when they are present in this with the proximity of exteins together to get this structure, they won't harm the cell. But whenever you get clipped out this intein sequence, because this is not required, whenever you decide that we don't want inteins, so just cut it away, now intein will go and kill you. That's the process. Now this concept is also telling us that the concept of placing inteins in between two exteins is a concept of a shellfish gene. So here the inteins is behaving like a shellfish. It's a selfish behavior is shown by here these inteins, because whenever intein is present, everything is fine, I won't harm you. Intein is telling I won't harm you, but whenever they cut inteins out, they just go and kill the cell. So the system is modified in such a way. But for eukaryotic system, you all know that we also have another. So let's consider this as a eukaryotic system. So if it is a eukaryotic system, it won't lead to death. Instead, what will happen? To remedy this double stranded break, we are having another same pair of DNA, same DNA, because we are having two pairs. So now what we can see, these two DNA can come together. Now here let's draw the structure. So here it comes, the double stranded break DNA, and here we get this whole DNA with our hand. Then what will happen? So I need to rub this part. So let me rub it here. Okay. So let me draw the double stranded break here. If you want to see double stranded, let me draw this with black color. Otherwise, it will be hard for you to understand. And watch this. So here it is, the double stranded break. Here the DNA without any break. Now what will happen here? To be very, very specific at this point, actually. Here it is, and obviously this one. So this is going to be the point, right? This is going to be the structure. This is how the structure is going to look like. So in those conditions, what will happen? You can imagine now, if I draw this structure, this upper strand is just go and replace this particular region here. So this strand just simply move up in this place, and these two strands just comes down to the level of this place. Now what will happen here, once these strands come really closer, in those case they will start forming this. Okay, and obviously, so what it will get is simply this. Sorry, it will be slight here. Okay, now what we get here, we get the desired gene. So here we get our gene, our DNA, double stranded DNA, which was previously double stranded. Here it will also be a new strand. So let me draw it first. Yes, and here it is. So now let me tell you, now once after this thing is done, once after everything is done, so if it happens in eukaryotic cell, what will happen? We create a double strand. Now these two are the pairs. So this is the from the set 1, and this is from the set 2. Okay, they are same DNA. So what we can, so this DNA are same from two different sets, one from father, one from mother. So we're having two set of every gene, every DNA. So what will happen? They go through this, which is called a double stranded break repair. double stranded break repair system. Now in this process they also utilize different enzymes, different ligase and different enzymes which will help them to establish this process, rec protein and other type of proteins. So finally what we get is our gene. Now what we get here, the double stranded DNA that we got here is also having a remedy copy from this new this old other set. So here this is the DNA which is getting which is getting a cleaved double stranded break. Now what we get, it is repaired, but what we get, both the DNA strands it is getting from the copy of it of the old one, right? So you get this copy. So that's how they make the repair in eukaryotic system. So if this thing happen in eukaryotic system, no matter to worry, but if it happens in prokaryotic system, it will kill the prokaryotic cell. Now this is the basic of the protein splicing. But another important thing I also want to tell you is that these protein splicing events are general in most of the cases in this general manner, but also sometimes these splicing events are a little bit more complex. For example, in case of blue-green bacteria like cyanocystis, like this bacteria cyanocystis. Now this kind of cyanocystis bacteria, and in those cases, in case of the cyanocystis bacteria, especially the protein called DNA-E, remember the DNA-E protein which is required for the replication purpose, this is called DNA-E protein for cyanocystis bacteria. Now in case of cyanocystis bacteria DNA protein, it is also having intein sequence like that in between the extein sequences. Now in this case we are having a different type of modifications. Now the modification we get in case of this cyanocystis DNA-E type of protein is having 2 introns and 2 exons, or 2 inteins and 2 exteins. So we simply get, this is, suppose this is our protein sequence. In this protein sequence we got here, say this is an extein 1 and here is the intein 1, this is an extein 2 and the intein 2. So suppose we are having extein 1, this is extein 2 and we are having inteins. So what we are having 2 exteins and 2 intein sequences, but they are displaced, or they are placed in distance, so gap, so in between there is a gap region also. So you can get this kind of things. Now what will happen and what kind of protein splicing will happen in this case of conditions where we are having 2 inteins, we don't require these inteins, we need to, so what do we make gap actually here? Gap means there are also protein coding parts, so no problem about that, but we need to get rid of these inteins, right? Because inteins are the segments which are not coding for any proteins, so you need to get rid of inteins. So what will they do in case of these inteins in this case? So simply they get another approach is simply to do these things differently. So they simply get the protein here. So here it is, the protein having intein and here exene and intein. So it is intein and this is obviously extein 1. And also what they prepare differently, they also prepare this extein 2 and intein 2. So get both these things together. Now what will they do? So they will recombine. And they will make a crossing. So this cross linking will help them to engage this E1 or extein 1 with extein 2 and this intein 1 with intein 2. So that's the process of getting this result in case of this cyanocystis bacteria DNA proteins. So we get this kind of elements from this segment. We get this. Now in this case we will only require this extein to be attached together, so individually they provide this extein 1 with intein 1 and extein 2 with intein 2. Then what will they do? They combine this exteins together to get the functional region of the protein, and also they combine inteins together to get the protein to be discarded. Okay, so this type of things can be seen. So it's a little bit complex. Now this part, this protein splicing, is a very new part and it is under active research. So I have found this part of the section only now. So I hope this video helps you to understand what protein splicing is and how it is done. Okay, so thank you.